we have ARMO 4021? Sorry, 5021. So we'll see this is November 1978, and this is Armour's application for a separate product license for its high-potency factor VIII. You'll see, please find enclosed 6 copies of Volume 1 and 2 of our submission for a product license for a new high-potency form of factor VIII to be identified as high-potency factor VIII. And then the second paragraph, reference to our previous submission, so that's the product application license we looked at before the break, will confirm that we've examined this product for quality and safety in a similar manner that previously agreed. And that application for a product license was successful, as we will see from ARMO 5036. This is the Licensing Authority's confirmation 13th of June 1979 that the authority had been given for the grant of a product license for high-potency factor VIII. And we'll see the separate license number there, 0231/0044. If we go over the page, we can see the license itself, and the date of grant is the 13th of June 1979. In terms of supplemental conditions or requirements, page 3 sets out what I think are largely the standard conditions of the product license at the time. So paragraph 1 refers to compliance with various statutory requirements. Paragraph 2 is leaflets shall comply with the requirements of the Medicines Leaflets Regulations 1977. Likewise, labels shall comply with the Medicines Labels Regulations 1976. Paragraph 3, the product shall not be recommended to be used for any purposes other than those specified in Part 1 of this schedule as uses. 4 is the specification shall be in accordance with the information contained in the application. And 5 is the product should be manufactured only in accordance with the method given in the application for this product license. So no separate or additional conditions appear to have been imposed in relation to this particular product. If we go to the application, which is at ARMO 0000, so 5023, and if we go to page 3, we'll see a similar form to that which we looked at before the break. So the name of the product is the high potency factor VIII. We go down the page to box 7, details of previous applications, references made to the factor VIII license. And then the very bottom of the page, we can see the date of the application, 20th of November 1978. If we go a further 2 pages on, please, we'll see there's some supplementary details in relation to product literature, labelling and package insert. Details are as attached. Data sheets, it's intended to defer final drafting until the licence is granted. And I'll come on to some aspects relating to labelling and data sheets in due course. But if we just turn over the page, we can see the draft package insert that was included with the application. And if we go to page 10, we can see it was proposed that the warning should read as there set out. Factor VIII is prepared from human plasma, each donation of which has been found negative for hepatitis B surface antigen by the radioimmunoassay method. In addition, each batch after reconstitution, as recommended on page 3, has been tested and found negative by the RIA method. However, since no completely reliable laboratory test is yet available to detect all potentially infectious plasma donations, the risk of transmitting viral hepatitis is still present. Just pausing there, and we'll see various other references to hepatitis B in very similar form to the references that were in the 1975 application, the original application for factor VIII. We're now, of course, in late 1978. All the references that we see are in the context of hepatitis B. There's the general reference there to viral hepatitis and that risk being still present. But there is nothing in the material submitted to the licensing authority that we've seen which addresses specifically any issues relating to non-A, non-B hepatitis, the existence of which, so you may think, was well known and understood by late 1978.
Well, yes, but the expression viral hepatitis it is capable of covering both. It
is undoubtedly capable of covering both. The question might arise as to how it might be understood by those for whom the warnings are intended in the context of a discussion about testing for hepatitis B and no such test to detect all potentially infectious plasma donations. Would that be understood by the reasonable reader or reasonable patient if they ever saw this as pointing out possible risks of transmission of non-A, non-B hepatitis? Within the same document, we can see page 12, bottom of the page, caution at the bottom that the product is prepared from pooled human plasma despite careful selection of donors and non-reactivity of the reconstituted solution for hepatitis B antigen by the radioimmunoassay procedure. Freedom from causal agents of viral hepatitis cannot be assumed. So the same terminology. And then the next page, last paragraph. High-potency factor VIII is manufactured from human plasma donations collected at licensed establishments, which are subject to USA federal law. So here, by this time, it's being said all collections are from licensed establishments. Each donation and each product lot has been found negative for hepatitis B surface antigen by RIA. Vials from the final dosage form are then examined for quality and safety aspects, agreed for the Armour product, and that's a reference to the original factor VIII, which was licensed for supply to hospitals 20th of March 1976. And then top of the next page, in terms of manufacture, all plasma is collected in establishments licensed by the FDA Bureau of Biologics and transferred to Armour under conditions defined in Title 21 of the Code of Federal Regulations. Plasma and plasma records are inspected upon receipt at Armour Pharmaceutical Company, Kankakee, Illinois, and if satisfactory, the plasma is approved for fractionation. And then if we turn to page 27, bottom half of the page, we can see there the plasma properties are set out, and 0.5 is free of hepatitis B surface antigen as tested on individual units of plasma by radioimmunoassay or other assay meeting requirements of Title 21. Over the page, on page 28, the bottom half of the page, so paragraph 3, there are various requirements there in relation to information to be included with shipments of plasma to Armour, so from the collection centres to Armour's manufacturing plant. And F is a statement confirming that all units have been tested and found non-reactive for hepatitis B surface antigen as defined in Title 21 Code of Federal Regulations. If we go on to page 31, there's a draft data sheet, and we can see the bottom of the page, sorry, top of the page, this is a draft data sheet for the plasma to be submitted with each shipment of plasma sent to Armour in Kankakee. And the bottom of the page, you'll see there hepatitis B surface antigen. So that's the data sheet requirement. And then if we go to page 50, we can see this is part of the quality control material submitted with the license application, similar to what we saw in relation to the 1975 application, hepatitis-associated antibody, biological principles of the procedure, and then there is a description of the radioimmunoassay technique that's used in similar terms to the terms we saw previously. So that's the application for the high-potency factor VIII, and as we've seen, the license was granted in June 1979. If we turn to ARMO 5046, We can see a follow-up letter from Armour to the Department of Health, 21st of November 1979. The text of the letter refers to a request that a copy of any data sheets be forwarded to the department. It is our intention to make this product available early in 1980, and in connection with this, we've now finalised the data sheet text. And if we go to page... Oh, sorry, I should just continue with that letter. Last few lines. We would like to point out that because of the specialised nature and use of the product, it is not our intention to include the text of this data sheet in the ABPI compendium. And we'll look at what was and was not within the ABPI compendium tomorrow. If we go to the third page, we can see the data sheet being sent to the licensing authority. And if we go over 2 pages, it should be to page 5, we can see under the heading warning, again, similar terminology to elsewhere in the documentation. Factor VIII is prepared from human plasma, each donation of which has been found negative for hepatitis B surface antigen by the RIA method. In addition, each batch after reconstitution as recommended has been tested and found negative by the RIA method. However, since no completely reliable laboratory test is yet available to detect all potentially infectious plasma donations, the risk of transmitting viral hepatitis is still present. And then it would appear, if we go to ARMO 5047, this is an application to vary the high potency factor VIII license. The basis of the variation is not particularly material, but you'll see the date is February 1980, and if we look at the first paragraph, last sentence, it says it's been our intention to launch the product in January this year, but to date the product has not been made commercially available. So there was a period of delay before Armour was actually marketed in the UK, although, as we'll see shortly, it did subsequently become available and was used. There's a product licence application also made for high potency factor VIII in Ireland by Armour. If we look at ARMO 5092, if we go first of all to page 5, we can see paragraph 12, the heading warning, again in very similar, potentially identical terms to the application that was made to the UK licensing authority. What's interesting in relation to this application is some of the clinical studies material that was provided to accompany it. So if we go to page 135, we'll see as part of the material that was included within the Irish licence application is a letter here from Dr. Levine, February 1978, enclosing trial data in relation to the product. And then if we turn to page 143, we can see under the heading Summary of Trials Reported, the following information summarises the 4 separate evaluations of high potency factor VIII. And then if we go towards the bottom of the page, we can see that there's a reference under the heading Adverse Reactions to one case of an adverse reaction being reported, thought by the clinician to be a case of short incubation non-A, non-B hepatitis. And over the page, there is what is presumably some details of how that short incubation hepatitis was said to have manifested itself. And then if we go to page 146, there is there a further description about a patient's, it's about 5 lines down in study 1, top of the page, one patient developing hepatitis 10 days after the infusion. made a prompt and complete recovery, showed no evidence of liver damage, and follow-up 1 month later. And then towards the bottom of that paragraph, it says no other adverse effects were observed in any of the 10 patients. So it's a short and passing reference to non-A, non-B hepatitis in the Irish application that was being made in around the same time. Oh, no, sorry, a little later. The clinical trial data was available in the late 70s, but it's a November 1981 application. And then if we turn to ARMO 40116, this is a letter dated November 1982 to the Department's Medicines Division. The particular issue is a request for a shelf life extension for high potency factor VIII, but we can see from this that by this time clearly high potency factor VIII is being marketed within the United Kingdom. Third sentence, this material is required urgently to fulfil commitments to our customers. So the precise date upon which high potency factor VIII became was being supplied within the UK is unclear, but sometime between 1980 and 1982. If we then turn to ARMO 40132, please. We'll see there in February 1984, Armour wrote to the Medicines Division at the Department of Health and Social Security. You'll see their reference to the 2 product licenses that we've looked at. And then the text of the letter says, further to today's telephone conversation, I confirm that the name factor VIII shall be changed to factor VIII heat treated, and the name high potency factor VIII should be changed to high potency factor VIII heat treated. Labelling will be amended accordingly. I'll come back to the question of the heat treated product shortly. If we just next look at ARMO 40145. This is a letter May 1984 in relation to the high potency factor VIII from Armour to the Medicines Division at the department, requiring a product licence after the expiry date of the existing licence. And if we go over the page, we can see the renewal application there set out. For reasons that are unclear, that the change of name to heat-treated factor VIII, well, is not there incorporated. So this appears to be an application for renewal of the non-heat-treated high-potency factor VIII product licence. And if we go to ARMO 40153, we will see that it was indeed renewed on the 13th of June 1984. The product licence for the high potency factor VIII there renewed. But before I look a little further at the question of the heat-treated product, I just want to show you some documents which cast some light on the extent to which Armour occupied a significant market share within the United Kingdom. So if we go firstly to DHSC 0003719_118, this is a table from the 21st of or dated the 21st of December 1976 within the department's supply division, addressed to Dr. Waiter, and its sales figures for October, together with total figures for the contract year. And we can see there that the Armour occupy a fairly small part of the market at this point in time, because, of course, factor VIII had only been licensed earlier in 1976. So we can see the total for the 12 months to the end of October 1976 for Armour was 897,308 units. Travenol and Immuno, which had both had licenses for longer by that point in time, much larger amounts. Abbott, a smaller amount, 383,624. So that's the position in the early days. If we then go to PRSE 0003,437, we will see the position changes over the following years. This is a table showing quantities of factor VIII concentrate used in UK haemophiliacs breakdown by manufacturer for the years 1980 and 1981. And you'll see from that that Armour are by far and away the largest supplier in terms of quantities. So 1980, 16,576,000 international units, and for 1981, 14,646. So 14,646,000 units. And then the figures speak for themselves. The next largest is, I think, Immuno's cryobilin product, and then Highland's Haemophil. The annual returns, I'm not going to go through details of annual returns, but we've looked at the annual returns for some of the bigger centres and some of the reference centres to see if there are any particular patterns discernible in terms of use of Armour products. The short answer is that it varies enormously from centre to centre. So we can take that down, thank you, Shumik. So I'm just going to take a handful of examples without pushing up any of the underlying documents, many of which in terms of annual returns we've looked at in the course of evidence and presentation so far. If we take Birmingham Children's Centre, Armour is first used to a modest extent in 1977. Its usage increases over the years that follow until in 1980 it's around 1 million units of Armour used. And by 1981, Armour was the only commercial concentrate in use at Birmingham Children's Hospital, with the figures being around 1.4 million in 1983 and around 1.6 million in 1984. That you'll recall is the centre of which Dr. Hill was the director. A rather different position. The picture emerges in relation to Belfast. Armour's first used in Belfast in 1980 to a relatively modest extent. Increased usage in 1982. It was the commercial concentrate used second only to cryobilin in terms of volume, but and then a significant usage in 1984. We don't have the returns of Belfast for 1983. Cardiff, by way of contrast, which of course is the centre of which Professor Bloom was the director, shows for the most part fairly limited usage over a number of years in the late 70s of Armour, until around 1982, when it's used to a greater extent, but amongst a range of commercial concentrates they're used, and then barely used at all in 1983 and 1984. Interestingly, if we look at one of the other children's hospitals, so Glasgow Children's Hospital, it is apparent that Armour was the commercial product of choice for the then director, Dr. Willoughby. It's very substantially used from 1978 through to 1982. But the picture essentially varies very much from centre to centre, so there's no obvious pattern in terms of usage, other than, as the overall figures show, it, if not dominated the market, was a very significant player in the market by 1980.
And the reason for that?
There is no obvious reason that we can detect, I'm afraid. There was the issue relating to price that we saw referred to earlier in the 1970s in the departmental material we looked at before the break. The extent to which that price differential was maintained over the following years and the extent to which that in turn influenced individual centre directors is unclear. Unfortunately, for many of the centres, we don't have the relevant director alive to ask.
No, we do have, I suppose, a division, do we, in the one of those figures that you showed me earlier. Was it the 1976 table? That probably.
was DHSC 000.
118.
C719.
C719 underscore 118.
That's the one.
Where we've got the price there, and Armour is obviously selling for a fair bit less than both Travenol and Immuno. It's easy to work it out, because if one takes reads across to the figure on the right-hand side, having divided by 10, it's true both Travenol and Immuno are charging more per unit than the Armour is.
Yes.
Armour is charging less than 10p a unit.
Yes, and we saw that being a feature that apparently attracted itself to the department's supply division when the licence application is being considered. What I don't know, sir, is whether that
whether it was
maintained.
We don't have any other documents with values written on them.
Not off the top of my head. We might do. We can certainly see if we can find out any more about that.
Yeah.
There may be a range of reasons, of course, why directors choose particular products. We'll get a sense, I think, when we look at Bayer, perhaps, of some of the marketing techniques that were used, and it may be that Mr. Bishop will be able to cast some light on the issue as well, in terms of his interactions with haemophilia centre directors.
So at the moment, we don't know why it is that Armour began to conquer the market to the extent that in the figure you showed me from 1980-81, it was very nearly half of all the commercial concentrates used, and about 3 times as much as its nearest rivals. And it may be price, but that's the only factor that we can identify as a possible, and it's only a possible factor.
Yes, and it's the reason why I went back to some of the annual returns for the reference centres or bigger centres to see if there was any discernible pattern, but there wasn't. So, as I say, one sees there are some directors for whom Armour was plainly the commercial concentrate of choice, and Dr. Hill at Birmingham Children, Dr. Willoughby at Glasgow Children are 2 examples of that. But when we look at some of the other big reference centres, Cardiff, Newcastle, there isn't an obvious pattern that emerges in terms of an overwhelming preference for Armour or indeed any particular concentrate year after year.
Yes. Right, thank you.
If we then just pick the picture up in 1983, in terms of usage, DHSC 0002229_055. This is a minute. I think we probably looked at it when Dr. Wolford gave her evidence, but the present purposes, it's the table on the second page that is of interest. So it says the following companies import coagulation factor concentrates into the UK from the USA, and then we've got the companies listed, and then annual sales, and you'll see the figures in relation to factor 8, 15 to 20 million international units, and then the high potency factor 8, 1 to 1.5 million international units. Alpha's Profilate is 5 million. Travenol's Hemophil is 8 to 9 million. Immuno's Cryobulin, well, there are the various different figures. We've got the red pack, 0.9 million, the blue 4.1, and then the Prothromplex and the Fiber. And then Miles Coate, which we'll look at, and when we look at Bayer, 8 million. Again, Armour, as at June 1983, which is the date of this document, so the figures may, for example, go up, I don't know whether they go up into 83 or whether they stop at 82, but Armour continue to dominate the market. But again, I'm afraid that data doesn't explain the reason why. So then just to pick up the picture in relation to the licensing process for the heat-treated factor 8, if we go to SHPL 5067_028, this is a letter to Immuno, but we know from another letter that it was sent in similar or identical form to Armour. So it's 26th of November 1984. It's from M. Duncan this is to Immuno, but it says, following our recent telephone conversation, may I confirm that the licensing authority wishes to encourage all companies involved in the production of factor 8 to use a dry heat treatment process in the course of manufacture. We're inviting each company to consider this proposal and hopefully to make early abridged application for a new product license. And then if we go to ARMO 40156, we can see a letter from Armour, 4th of January 1985, application for variations to both product licenses. And then the letter begins with reference to the letter from Dr. M.E. Duncan, dated November the 26th. So clearly a letter in identical form or similar form went to Armour. We enclose applications, so let me read the whole thing. With reference to the letter from Dr. M.E. Duncan, dated November the 26th, 1984, and the undersigned, that's Mr. Ormers Collins, Regulatory Affairs Manager for Armour, meeting with her on December the 19th, 1984, we enclose applications, 2 copies of each file, to vary the above product licenses. The variation relates to the inclusion of a heat treatment stage in the processing of the products. Much of the information presented has already been assessed by the DHSS in relation to our CTX, so clinical trial exemption, and PL applications relating to non-A, non-B hepatitis. It is understood that such applications as these are being considered a priority by the licensing authority in view of the general situation relating to AIDS in the UK. So that's the application for variation for heat treatment being submitted by Armour in January 1985. If we go to ARMO 40157, we can see the application for the variation. This is in relation to the high potency factor 8, and the proposed variation we'll see from the right-hand side under the heading proposed. The method of manufacture will be as currently undertaken, but with an additional step involving heat treatment. The finished lyophilized vials from the current process will be subjected to a heat treatment in a water bath at an attained temperature of 60 degrees centigrade plus or minus 1 degree centigrade for a period of 30 hours. And the significance of that particular heat treatment method as opposed to others will become apparent when we look in more detail at the issue of seroconversion from the heat-treated concentrates in November. The application for the variation for the heat-treated factor 8 is at ARMO 40164. We can see it's dated January of 1985. And if we go to page 5, we'll see there the heading Reason for Change/Background Statement. For some years, work has been progressing at a number of centres, particularly in the USA, on the development of factor 8 products with reduced risk of the transmission of hepatitis. Work at Armour Pharmaceutical Company, Kankakee, Illinois, resulted in the development of a heat treatment stage incorporated as the final part of the process in the current production of intermediate potency factor 8 and high potency factor 8. Initial studies revealed that heat treatment, a commonly accepted concept in the destruction of heat labile viruses, was insufficient alone to prevent the transmission of hepatitis B, although evidence existed to suggest that non-A, non-B viruses might be removed by this process. The process was approved by the FDA for the US market and by the BGA for the German market. A product license application was submitted 16th February 1984 for the high-potency heat-treated factor 8. Earlier, a Ctx application was submitted for the intermediate purity product factor 8 heat-treated on the 11th of August 1983. in order to undertake a definitive study at UK haemophilia centres over an extended period of time in previously untreated patients to determine if infectivity of the product had been eliminated. Pausing there, we'll look at some documents relating to that Armour study of its heat-treated material either this afternoon or tomorrow morning. Subsequent events indicated strongly that while the products were clinically effective, the heat treatment process employed did not achieve the desired objective in relation to non-A, non-B hepatitis, and the product license application was withdrawn and the Ctx surrendered in November 1984. During 1984, concern among UK physicians continued to grow over the AIDS situation, and then reference is made to isolation of the causative virus and to the development of tests for the virus. If we then pick it up in the next paragraph, the heat treatment process developed for factor VIII and high-potency factor VIII products in relation to non-A, non-B hepatitis looked as though it may be sufficient to destroy the LAV-HTLV3 virus. A programme of work to assess the situation in relation to a number of heat treatment processes and products has been undertaken by the US Centre for Diseases Control in conjunction with the FDA Bureau of Biologics. It would appear that in the in vitro situation, 60 degrees for 4 minutes will inactivate the virus in certain conditions. And then over the page, there's then further information about the proposed heat treatment process, which was 60 degrees centigrade for 30 hours. It's asserted that all the indications are that this process is effective in the destruction of the virus. And then if we pick it up a few lines down in that first paragraph, there have been an unprecedented number of named patient requests, and the situation with regard to such a supply on the scale sought is totally unsatisfactory. It's clear that haemophilia centre directors believe that a heat-treated product, even though not proven over time to eliminate the AIDS risk virus, is preferable to a non-heat-treated product. And then the next paragraph explains that studies are continuing. However, we believe the products with a heat treatment process as proposed are safe and effective. Furthermore, we believe the heat treatment process proposed will render the existing products obsolete and thus apply for a variation to the existing licenses. And then there is a large amount of material which is provided. I don't think we need to look at most of it, but if we go to page 32, we can see some information about studies in humans of the heat-treated product. Heat-treated factor VIII proposed for introduction onto the UK market has been and is currently on the US and German markets. Hence, there is already considerable exposure of patients to this product. Furthermore, the product is being used in the UK on a named patient basis and has been used in the abortive clinical study, Ctx 0231/0070A. Additionally, a half-life study has been conducted in the UK by Dr. Ritzer, consultant haematologist at the Oxford Haemophilia Centre. And then there is reference to a half-life study in the US that was conducted. And then if we go towards the bottom of the page, we can pick up the reference to Dr. Ritzer's study. The second study, as mentioned above, was conducted by Dr. Ritzer, Oxford Haemophilia Centre, on the material used under the Ctx 0231/0070A. And it goes on to set out a conclusion in relation to the half-life for both heat-treated and conventional factor VIII. I'll come back to elements of the study, but if we then go to it's page 166. No, I don't want to conflate the US and the Oxford studies. I can pick up the Oxford study by reference to the Ctx material, because that may be a reference to... Oh, no, this is the Oxford one, I think. Sorry. Revlon Healthcare UK Limited supplied the investigator with sufficient supplies of the following to allow for completion of the study, and there's reference to 2 heat-treated factor VIII lots. standard commercially available factor VIII lot, and then the lot number given, taken from the stock at the Oxford Haemophilia Centre. And then there's a reference to 4 patients being entered into the study. And if we go to the next page, you'll see under the heading Informed Consent, it said written informed consent was obtained from all participating patients. And then the results over the page, page 168 are there set out. So that I think is really just an indication of one of the studies that was undertaken domestically in relation to heat-treated factor VIII prior to it being licensed, the half-life study. There's a further study that we'll come on to. Before we do that, if we go to ARMO 40301, you'll see that this is a letter from Revlon Healthcare / Armour Pharmaceutical Company Limited, 2nd of November 1983, to Dr. Preston in Sheffield. I confirm we've now been granted our clinical trial exemption certificate by the department and can proceed with trials to evaluate our heat-treated factor VIII to the protocol left with you for information. And then there's the reference to the study underway at Oxford. So we have a study in Oxford and a study in Sheffield ongoing essentially from 1983 in relation to the heat-treated factor VIII. And then if we go to ARMO 40121, this is the application, the Ctx application. If we turn to page 3, we'll see there that it's an application under the Clinical Trials Order in 1981. And if we go over the page to page 4, we can see the date of it, which is, I think it's the 18th of August, 1983. If we go to page 5, we can see a section, Particulars of Medicinal Product and Trial. And if we go over the page, we're told full details of the proposed trial are that the investigators, Dr. Ritzer at Oxford, the duration of the trial is 1 year, number of patients involved, approximately 25, depending upon availability of patients fulfilling inclusion criteria. And then if we go to page 12, we can see the protocol for this study. It's dated May of 1983, the date's in the bottom left-hand corner. And if we go to the next page, we get some background information about the trial. One of the primary concerns in the use of coagulation factors in the haemophiliac patient who has mild to moderate disease, which requires infrequent treatment, or in the newly diagnosed patient, is the knowledge that each exposure presents a risk of causing hepatitis. And then under the heading Objective, further down the page, it is the purpose of this study to use our specially prepared factor VIII product exclusively for an extended period of time in a number of previously untreated patients or in those who have received minimal treatment to determine if infectivity of the product has been eliminated. So this was a proposed PUP study, effectively. Minimal treatments defined as having received no concentrate or cryoprecipitate during the preceding 6 months and not having undergone major surgery, etc., and having no history of hepatitis, yellow jaundice, subclinical hepatitis, or any abnormal liver function tests. And then if we go over the page, we see the study design. Selected study sites will be haemophilia centres run by recognised experts in haemophilia care who have an adequate number of patients to assure the recruitment of at least 5 untreated subjects each over a 1-year study period. In addition, these centres will be asked to recruit an equal number of patients who've had infrequent treatment and are free of hepatitis markers. Next paragraph, it will be essential for the centres recruited to have close control over their patients to ensure that those entered into the study have access to and use only the trial factor VIII. Any break in this rule will end the study for that subject. At the time the non-study product is used, patients will be entered into the study as they require factor VIII HT, heat treated. The end point of the study will be the presence or absence of hepatitis as measured by hepatitis markers and liver chemistries taken serially over the 1-year period of study. Top of the next page, all patients or their guardians will have the purpose of the study carefully explained and will sign an informed consent. They will understand and agree to the use of study factor VIII exclusively for the 1-year period of the study. However, in the best interest of their patients, the physicians may prescribe any treatment considered necessary. If this includes factor VIII other than the study material, the patients will continue to be followed, but not included in the analysis. And then if we just go over to the next page and pick it up under study design towards the bottom of the page, this is the penultimate paragraph, the patients will be mild haemophiliacs and therefore not on home treatment. Any intercurrent illness will be recorded with date and time of such illness. And then, bottom of the page, the patient records or duplicates will be segregated at the treating institution to ensure that the investigator or staff will be notified at any time the patient enters the institution, either as an outpatient or inpatient. Top of the next page. In the absence of transfusion hepatitis, patients will be followed for 1 year following treatment with heat-treated factor VIII. And then it explains about undertaking of liver function tests and other tests, collection of blood, etc., etc. The study will continue for 12 months. A complete physical examination will be repeated at the 12-month visit. Those patients whose liver function tests remain elevated for 1 year after the attack of non-A, non-B hepatitis or become carriers of hepatitis B virus will be referred to the local liver clinic for investigation of chronic liver disease. Liver biopsy will only be carried out if clinically indicated. And then one final reference. Is at page 21. Now this is not part of the protocol for the study that we've just been looking at, but this appears to be part of the same material that was submitted to the licensing authority in relation to the Ctx application. So application under the Medicines Exemption from Licenses Clinical Trials Order 1981, factor VIII heat-treated product. We can see the first paragraph talks about the time spent to on trying to develop a factor VIII with a reduced risk of transmission of hepatitis. And then if we go, however, to the penultimate paragraph, it says this: in addition, the recent upsurge in incidence of acquired immune deficiency syndrome has highlighted the problem of possible transmission of illness through blood products. The nature of the AIDS syndrome and lack of knowledge of the background etiology are such that it is impossible to determine whether procedures such as heat treatment might afford protection. Nevertheless, there's a growing body of opinion that heat treatment of material could maximise safety without detriment to the product or its clinical efficacy. The present study is proposed to assess the value of heat treatment on transmission of non-A, non-B hepatitis. The discovery of common interests led to an approach to the UK Haemophilia Centre Directors Working Party on Hepatitis, with resultant collaboration in the design of the study. Top of the next page. The Haemophilia Centre Directors expressed a wish that the study be conducted with factor VIII rather than high-potency factor VIII. As for economic reasons, that may be the answer, sir, to your question. This is the product most commonly used in the UK.
Well, it may only be the difference between the people choosing the cheaper of those two, as opposed to Armour as against the rest.
Yes. And then the next paragraph, this situation has been discussed informally with DHSS professional staff. It was agreed that this present application could be supported with development data on the high-potency factor VIII product, etc. We believe that this study, performed under the auspices of the country's leading haemophilia specialists, studying in depth a group of highly investigated patients, is the definitive course to follow at this time in order to prove the efficacy of heat-treated factor VIII in the prevention of transmission of non-A, non-B hepatitis. So the purpose of going to that in some detail was really just to illuminate the protocol drawn up by Armour together with, it would appear, haemophilia centre directors, Dr. Ritzer being identified as the investigator, contemplated in 1983, with the knowledge of the risk of AIDS one sees referenced in the page we just looked at, the use of heat-treated factor VIII in patients with mild haemophilia.
Yes, patients who had not previously needed treatment with any factor concentrate.
Yes.
And so if they were to be enrolled in the study, assuming it, well, I don't think one can assume it to be ethical, given what our ethicists have said about the need for therapeutic benefit for a patient. But if it were, they'd have to be told about the potential risk of AIDS as well as potential risk of hepatitis coming from a treatment they'd never had before.
Yes. Yes, the ethical questions to which the protocol and study give rise are multiple and fairly evident.
Yes. Is there any material, since we're on that particular tack, is there any material that you've spotted in the protocol which suggests that there would have been treatment in any event with a non-heat-treated factor product?
There was one passing sentence that is all I can recall, which if we go back to page 14 of the document that we had out, ARMO 40121. If we look at the second paragraph under study design, and it would be reading a lot into the use of, into one short sentence, it's the last sentence, patients will be entered into the study as they require factor VIII HT. It could be said that the inference to draw from that is that they're only treated if they require the product, but I don't recall there being anything else which addresses that issue. I'll reread it again overnight.
I mean, that may be of some importance, but I mean, that could indicate that although one starts off with a recruitment of 20-odd patients who've never been treated before, there will be an occasion or so when one of them needs to have an agent to stop bleeding and assume that DDAVP or won't work if it's one of those cases, or cryoprecipitate is thought insufficient, that they might then have used whatever factor product was available.
Yes, there are quite a lot of assumptions that one has to build into that one sentence, and it's perhaps instructive to note the date of this protocol is May 1983.
Yes.
One need only think of the consideration that was being given by the Haemophilia Reference Centre directors at their special meeting in May 1983, resulting in that letter from June of 1983, which talked about the position of those who were previously untreated patients.
Yes.
But that, sir, is probably an appropriate note on which to end for today.
Yes. Yes,
But
thank you.
I should say, it's possible that I may finish the Armour documents in the course of tomorrow, because there is so much more of the Armour story that we're going to be telling in November. If I do, then we may go on to Bayer at some point in the course of tomorrow afternoon. Of course, you know that the time estimates from Counsel are hopelessly unreliable, so it may not turn out to be the case. But it's a possibility we might start Bayer tomorrow afternoon at some stage.
Yes, thank you.